I'm very happy to be here. I'm Niall Lennon from the Genomics platform, if you haven't seen or met me before. And Chris had asked us to come and start in this primer series, talk about, just give an update where we're at with the technologies that are being used at Broad and other ones you might have heard of and might be curious about, where they're at now, what the plan is. And so Maura Costello is a senior scientist in the genomics platform, has been here for many years looking at technologies and really has a very deep understanding of all of the technologies we've used, and continues to look at new technologies as they come around and as we think about their application to the science that happens at Broad. So with that, let Maura take it away and really give us an introduction and tell us where we're at. Please feel free to ask questions. It is being live streamed, so we'll repeat the questions up here for anyone who is remote. Thank you.
Great, thanks, Niall. There is going to be time at the end of the talk for Q&A, but if you have a burning question while we're going through the slides, just feel free to raise your hand, that's fine. As Niall said, my name is Maura Costello. I've been at the Broad Genomics platform for, it was 12 years in June. So there's people at Broad who've been here a lot longer than that, but I came in, basically my first week was the first week that the old Solexa machine, which would become the Illumina sequencer, showed up. So I had pretty good timing, basically being able to see a lot of the technologies as they developed. So sort of walk through kind of where as Niall said, where we are now, and then kind of where the genomics platform is sort of looking for the near future, basically, where we want to be going. So right, that's basically what I just talked about. So talk about right now, we're mostly doing what is called short reads. So that's Illumina sequencing, basically, as you guys know it. Where we're going next is on the shorter term sort of development path is looking at more long reads technologies, and I'll talk about that. And then again, there'll be time for more detailed Q&A if needed. So Illumina sequencing, that's obviously where we are right now. It's currently in italics because it won't be the only sequencing technology forever at the GP, hopefully. But that is currently the only sequencing technology that we have available to offer sequencing to the Broad community. And it's because Illumina is far and away the industry leader. So they have 70% of the market share. When people talk about sequencing, 9 times out of 10, they're talking about Illumina. Their technology incorporates sequencing by synthesis, SBS, that's sort of their patent. But it's limited to short read sequencing up to 300 base pairs typically. So typically we do a paired 2x150 base pair read for folks. Some of their instruments can go as long as 500, but you get much less data on the back end if you go that long. So just a real quick refresher, you guys have probably seen this many, many times, but how Illumina works is basically take your DNA, modify it by adding ligation adapters to it that are complementary to sequences that are on the surface of the flow cell. Flow cell is basically a microscope slide for the most part, the surface of which is covered with grafted oligos that are complementary to the adapter oligos you added to your fragments. And then there's multiple amplification steps. I won't go through this too in detail, but you end up basically copying your DNA fragments to the surface of that flow cell, sort of amplifying them up so you get a good signal. And then the sequencer, the sequencer can read those clusters, we call them, which are sort of the clumps, clonal clusters, I should say, of the original pieces of DNA that you amplified. So there's 2 technologies that Illumina has. They're sort of, I say older technology, but they still sell instruments that do this, is the random flow cells. So they just have a sort of random lawn of primers. Basically, imagine like blades of grass in a field. And the DNA clusters just grow kind of willy-nilly. You can see they're all different sizes, all different shapes. They sort of grow into each other. The newer technologies, the HiSeq X and the NovaSeq, have these pattern flow cells. So the actual surface of the flow cell has these little nanowells in them. So it can actually sort of constrain those clusters, and you end up more of a honeycomb pattern. So you can really cram the data in, and it's much easier to sort of tell the clusters apart and get higher quality data. So we have machines and we still run machines that do both of these sort of modes. But regardless of sort of how the flow cell is set up, the sequencing happens exactly the same way, the actual sequencing on the instrument. So you've got the clusters on the flow cell. you're basically going to add, you linearize them, so they're all single-stranded, kind of all standing up at attention, all the pieces of DNA. And then you just add one fluorescently labeled base at a time, fire lasers, figure out what cluster has what base at each cycle, quench that fluorescence, and start again. And so we can do that for, like I had mentioned previously, most Illumina sequencers can do 300 cycles of this chemistry before it starts to lose quality. So that can be, typically it's broken up into 2 shorter reads, one on each side. So a 2 by 150 is sort of our standard read length. And so I mentioned the NovaSeq. So that's the newest sequencer from Illumina. And they kind of went with a bigger is better philosophy. The actual instrument is bigger. It sits on the floor. It's tall. It's almost as tall as a small person, basically. And the flow cells are much bigger as well. So these new instruments can produce 3 times as much data as the HiSeq X in about the same time. It might even be a little bit shorter. You can see the flow cell is actually bigger. So the one on the left is the HiSeq X flow cell. It's a delicate little microscope slide. You can see this guy's just like manhandling the NovaSeq flow cell. It's this big honking thing. So it's bigger than an iPhone Plus, but that way it just gets more data that way. It's just they went with, everything's bigger. So that's kind of their new flagship sequencer. I'm sure every few years they come out with the new greatest thing. So I'm sure in a couple years we'll be talking about something else. But for now, the NovaSeq is kind of their state-of-the-art technology. And I do want to say a quick aside about so sort of talked about how the sequencing chemistry kind of works the same regardless of the flow cell type. But there are a couple of subtle differences. So the new NovaSeq uses a different dye chemistry. So the original Illumina machines used 4 dyes. So each base had a different color. And so when you fired the lasers, they would light up a different color, and it was much more straightforward to say what base is what. The NovaSeq, to sort of save costs and to save time, they use a 2-dye approach. So it's sort of a combinatorial and lack of dye situation as well. So if you see here, so in this example, the T base has a green dye on it. The C base has a red dye on it. A has both dyes, both fluorophores, and G has 0 fluorophores. So when they fire the laser, they basically look to see where the bases are lighting up. So they have 2 lasers, a green and a red. So the C is going to light up very strongly with the red laser, and the T is going to light up very strongly with the green. A is actually going to light up a little in both because it's got both fluorophores, and G is going to be dark in both lasers. So that is a little interesting because especially if you've got, say, it's not, say, a human whole genome where there's a lot of base diversity. If you've got some kind of an amplicon or something that has a lot of Gs, you're going to have a lot of dark bases. So sometimes there's some issues. There can be issues, although they've worked on this, and this works surprisingly well, that it's hard to tell if there's just a lack of a signal or if there's actually a G base there. Like, is there no DNA there, or is there a G? So although this does work surprisingly well, we get, we're happy with the NovaSeq data. But because of this, it has slightly different biases than the old data. The sort of GC content, things like that are a little bit different. So we actually don't suggest mixing data from the two. I mean, it's, just because the data and the filtering that you do sort of on the back end are different enough that, especially if you're doing variant calling, if you're trying to do a joint call set, it might sort of introduce some error there. So in general, if projects have started on, say, the HiSeq X, we sort of say finish on the HiSeq X. If you start on NovaSeq, keep going on NovaSeq. So that's kind of our little tip. But so this is a lot of words, and sorry, I just realized that looking at this. But basically, this is just to say that we offer a lot of different flavors of sequencing right now for Illumina at the Genomics platform. So these are all available through the walk-up service. If you guys have used that before, it's basically through walk-up sequencing at the Genomics platform, we can sequence any sample you have, basically. We don't have to do the library prep. You do whatever you want, whatever customized special process you've got going, and we will deliver you sequencing data. So we've got basically all sort of sizes and flavors from MySeq, which is like sort of like the baby bear sequencer, where you get a small amount of data, but it's a very quick turnaround. And it's very customizable because it's just one sample per flow cell. So you can mess with things, custom primers, things like that, much more easy. All the way to the NovaSeq S4, which is giving you 1.5 billion reads per lane, 6 billion reads per flow cell, so, and then a bunch of different things in between. So, so again, we've been spending a lot of time the past few years focusing on short reads and our short read offerings. So this is sort of the summary of that. But if you're interested in doing any walk-up, I think you can email walkup@broadinstitute.org or genomics@broadinstitute.org, and we'll be able to help you out. It totally depends on there's a lot of different ones because of the read links and the cycles and things like that. I mean, we're in the hundreds of thousands of ranges per lane. It just depends on what you're doing. Yeah. And then a little aside on sort of a supplement to short read technology. So something we do have in the genomics platform available. There was sort of a flurry of interest in it maybe a year or two ago, and now it's kind of died down. But linked reads with 10x genomics. So if folks are familiar with single cell, you hear a lot about 10x for their ability to do single cell sequencing. They also can make whole genome DNA libraries, not from single cell, but just from genomic DNA. Using that same technology, instead of tagging all the reads to say they were from the same cell, they can tag reads saying they were from the same large original sort of genomic molecule. So, you can basically make emulsions with these sort of oligo tags. They get added to they get amplified copies of the original long, 100 or 200 or however long your DNA is, KB DNA. You then break those emulsions, and now you have copies tagged from the same molecule with the same tag, basically. So you can then make Illumina libraries out of those, sequence them as normal, and then use 10x's software to sort of rebuild those long molecules based on the tag. So not quite long-read sequencing, but it provides you at least some information that these pieces of DNA were all on the same original fragment. So I mean, the pros of that is that we have it now. If you did want to do a project, we could accommodate you. The cons are, we've heard from users that because there's multiple amplification steps, that you end up with some gaps and some biases. So, but again, that is something that we have available for folks if interested. So that's pretty much all of our kind of basic Illumina offerings. I didn't want to spend too much time on that. But then, just to sort of talk about technologies in general, there are other short-read technologies out there, just not physically here, but they are out there in the world. So I just wanted to spend a couple minutes just talking about those, just to sort of point them out to folks. So the two that I sort of hear most about are the Thermo Fisher Ion Gene Studio S5, which used to be called the Ion Torrent, if anyone has ever heard that name before. So they, these machines are still, in fact, this Gene Studio S5 is their new version they just released this year. They have small chips, and this is done with semiconductor sequencing, which means instead of using dyes, when the base is incorporated, they measure the sort of the pH change, basically, from the base being incorporated, and they sort of record that is the base. So that's kind of how it works. It's in theory, it's a much cheaper chemistry to run. However, you get less data typically than you do with Illumina. And also folks have reported that there's higher error rates than Illumina. But they definitely still sell these instruments, Thermo Fisher. The other sequencer, this is a kind of interesting one, is the QIAGEN Gene Reader. So they're a little bit cagey about exactly how this works until you buy one, apparently. I tried to find a lot of information about it, and I couldn't. But QIAGEN has basically made an end-to-end solution. So they want to sell you this package of instruments that includes basically an entire end-to-end workflow, from raw material, like a blood sample or a tissue sample, getting the machine. Actually, if you look, this machine on the far left gets the DNA out of the material. This machine here prepares libraries. It's a little automated library making machine. And then here's your sequencer. So these guys are mainly targeting the clinical space. And again, you don't get a lot of data out of this. This was based on a report from last year that folks were seeing around 5, a little more gigabases per run. So again, not a lot of data, but ideal for a lot of targeted panels. And because it's an end-to-end solution, it's probably pretty good for clinical labs, especially small clinical labs that maybe don't have a lot of infrastructure support. So, and I'm sure there's been a ton of other technologies that have come and gone. But these are the 2 sort of short read technologies that are still sort of lasting and are still available for folks to purchase. And then the other technology, because there is another, is so Bgi Sequencing. So they do not sell a box. You can't buy their sequencer. They sell a service using their proprietary sequencing technology. So how it works, so this is based on, for any sequencing nerds, there's a company, Complete Genomics, that came up with this technology, and then Bgi acquired them. So they use DNA nanoballs. That's sort of their thing. So you start by circularizing your DNA that you want to sequence. You make single-stranded circles, or double-stranded, and then you denature those circles. You then do a rolling circle isothermal amplification. And if anyone ever did Sanger sequencing, it's very similar to the T-Phi, the Templify reaction for 529 that we used to do way back in the day. So you end up basically making a bunch of sort of repeating copies of this original circle, and it all kind of makes this big ball of DNA, basically. So that's a nanoball, and they kind of sort of self-assemble, and you end up with these sort of almost like in-solution clusters. And then somehow, and I don't really know how because they don't really tell you, you seed these nanoballs onto a surface of some kind of a flow cell, and then those get sequenced. And so they offer this technology as a service. They have PCR-free options available, which basically means there was no PCR prior to this circularization step. And so that's kind of how their technology works. And again, there's a little bit of like a hand wave because some of this we don't know how this really works because it's proprietary. But and they have a few different sort of models and different for kind of how we offer different flavors of sequencing. They also do as well. And so they've also been saying that they can do a $600 human whole genome, which is very, very cheap considering if you know about how much genomes cost typically. So, we sort of have been hearing reports of this. So we wanted to look into what is their $600 genome and what do you get? And so we just, we were curious. So, so the first question is, well, what am I getting for $600? So Bgi, they only guarantee 90 gigabases of raw data. They don't guarantee that you're going to get a certain coverage of the genome. It's just, here's 90 gigabases. They don't guarantee a quality. They just say, here it is. So one thing that we've sort of been looking at, and again, this is a different library prep method, but still, typically 90 gigabases, we haven't seen it be enough for a 30x genome. I mean, if you think that the human genome is 3 gigabases and a little bit more, 90 gigabases is like literally the bare minimum, and it's probably not going to be a true 30x genome. We guarantee a 30x, so it's a little plug for us basically, but we guarantee you at least 30x. We usually average higher than that, and we guarantee quality, evenness, that's deduplicated, all kinds of different measures. So that's something to keep in mind. The other thing is, so they've released some data, actually. So they put up on the NIST, sort of genome in a bottle page, they've sequenced a lot of sort of HapMap trios and other samples on various technologies. So they've actually put up, and this was with Chris Mason at Weill Cornell. He sort of worked with Bgi, and they sequenced the NA12878 genome in a bottle reference and put that data up. So we've been able to download it. I think folks are still looking at the nitty-gritty of the data, coverage and sensitivity and things like that. But the one thing we did have real quick access to, because they gave it to us, basically, was the GC bias curve. So if you're not, it sort of looks a little light on the screen. If you're not used to looking at these, basically, this line at 1 here, basically this is plotting normalized coverage versus the GC content window. So this is real high AT, low GC, and this is real high, it's basically 100% GC. So in a perfectly even library where there is no bias, you would just see coverage on a perfectly straight line across 1. we never really see that, and there's some like weird artifacts in the analysis where we always see these little bumps. But for the most part, our PCR-free genome, it dips a little. We actually overrepresented high GC, which is interesting, but for the most part, we dance right around this nice 1 line here. And this is hard to see because these points are red instead of blue, but the BGI one basically starts way down here, kind of levels out, and then kind of dips again. So they seem to have some GC issues on the extreme ends, which is indicative of there's some kind of an unevenness happening. So again, it's like a buyer beware thing, but we just wanted to point that out. This is another technology that is available to people, but it may not give you exactly what you're used to seeing when you think about a standard genome.
So one thing also, just to, as Maura mentioned, we are looking more at the data to understand genotype concordance, sensitivity, just to understand the data. If you are looking at the data and you want to compare it yourself, like Maura said, it is available. They've put it up on a FTP site where you can retrieve the 12878 data and you can look at it. Just be careful that, like Maura said, the deliverable that if you order a genome from them is 90 gigs, which is about 28x of a genome. The demo data they've released is a 50x genome, so it's not exactly what you would get. So we have to downsample it to actually understand what it would look like at the coverage you would get. Just, again, some interesting practices.
But yeah, and also if you've done some of your project with Illumina genomes, it might be weird to mix the data because they're different machines. So it's just something to keep in mind. So. So that's basically what sort of the state of the art now, short read data. Next, I kind of want to talk about, so what we're exploring right now, long read sequencing technologies is sort of our current sort of development plan for sequencing. So some of these technologies aren't really new. It's just they're a little bit new to us, and or they've sort of had some improvements in the past year or so that have made them more attractive for us to use. So just a quick aside, and so what can long reads provide? So short read data is great for variant calling for single nucleotide variants, and you get a lot of it, and it's relatively cheap, and that's great. But a lot of times you can miss things with those short reads. Structural variation in particular, big deletions, big insertions, sort of duplications of chromosome arms and versions, translocations, all kinds of things that if you're just kind of putting like the jigsaw puzzle of little reads together, you might not realize is happening. Whereas if you had a long read that could span these whole sort of large rearrangements, you'd be able to see that better. So that's sort of why it's attractive to sort of pursue long reads. Oh yeah, and this is actually data from PacBio, which is I'll talk about in a second. It's a long read provider, but they basically showed that compared to various like either using optical mapping or various short read technologies, that them in blue, they get more sensitive to deletions of multiple sizes, even small ones, and even more so for insertions. So again, this is their data, but it just shows that these long reads can improve your project, especially if you're doing some rare disease or if you struck out on exome or genome. Maybe turning to long reads could get you more information.
Maura,
Yes?
why does 10x do worse than Illumina short reads?
I don't know. Again, this is PacBio's data, so, and 10x is a competitor, so I'm not sure why.
They also could be looking at here using 10x data natively without adding any short reads to it, where in some cases people combine short and long reads to actually maximally have sensitivity, and with 10x, because of the unevenness, you can have a harder time if you only use that data in isolation. But again, I think Maura's right. I think it could be some brinksmanship.
Yeah, I mean, take that with a grain of salt. But just basically, this is just illustrating that you can get more out of your projects if you do sort of incorporate long reads into it. So speaking of PacBio, so the PacBio Sequel is a long read sequencing instrument. And PacBio isn't a new company. They've been around since 2011. At 320 Charles Street for the genomics platform, we had the original PacBio, which was this enormous, like, I don't know, 10, 12-foot long behemoth that needed a concrete subfloor. It was like multiple tons. It was insane. Luckily, they have sort of streamlined their process over the years. So the Sequel, it's still large, it's still as tall as a human, but much smaller, and it doesn't need a concrete floor, so we can put it anywhere, which is nice. but right, it's not new to the Broad, and in fact, the PacBio moved to the Broad Technology Labs a few years back, or the former Broad Technology Labs. So it's not even new to the Broad. We've had PacBio technology for a bit. So we're actually now transferring the instrument back to the GP. So it will be, hopefully, an offering for walk-up sequencing probably next year at some time. So that's something to keep in mind. And they've also made some improvements to their technology that might make it more attractive for folks wanting to do human sequencing on it. The data output previously was sort of on the lower side, so it was more tuned for metagenomic or long transcriptome sequencing. Yes, they've made some improvements. So I just want to point out we are literally in the process of like crating it up, moving it, training people. So we won't be accepting any new projects on it until early 2019. So for folks who do use PacBio, I just wanted to lay that out there, that it literally is being decommissioned, put in a box, and shipped a few blocks. So that's going to take a little while to get it set up. So apologies for that, but we will be hopefully turning it back on early 2019. But when we turn it back on, this is how it works. So they use a technology called smart sequencing. So it's single molecule real-time sequencing. Slightly different than Illumina. So, well, not that different at the end of the day. So they, instead of with Illumina where you tether you sort of stick the DNA to the surface and then there's floating polymerases that kind of bind and amplify, they actually tether the polymerase to the surface. And you sort of feed your long molecule, single-stranded molecule, into these zero-mode waveguides. They're basically the little compartments that have the tethered polymerase. And the polymerase, oh, that looks like a Pac-Man there. Polymerase is adding these labeled nucleotides, and it's taking sort of, they call them movies, actually. It's taking sort of the images of each base being added and recording that fluorescence. And the current, on the current technology, there's up to 1 million of these zero-mode waveguides per smart cell. The smart cells are small. They're these little guys, and you can run a bunch of them sort of in a row. And another trick they do is their libraries use these, very similar to Illumina. You ligate adapters on the end, but they're these barbell adapters, these sort of semicircular adapters. And so when this gets denatured, this makes it look like it's the single strand, but it's actually circular. The polymerase can read around and around and around and around and around. So it can improve its accuracy because it can see the same base over and over again. And so you end up doing what they call consensus sort of base calling, where if you've seen this base 3 or 4 times, you have more confidence that's the true base. And their read lengths can be, or their fragments can be 2 kb to 30 kb or even more. So again, you get these really long reads that can help you kind of resolve some structural variation things. Oops. And so they've made some great updates. So the upgrade that we're doing now as we're moving it is they're calling it the Sequel 6.0 with new chemistry 3.0. But essentially, it increases the read length, so the amount of time that polymerase can keep going around that fragment to 100,000 base pairs. So if you've got a 10 kb fragment, you're going to be able to see that every base in that 10 kb fragment a bunch of times if you can go 100,000 times around, or 100,000 bases around. So that enhances the accuracy. And they're saying, and again, we have to look at this data ourselves, they're saying that now this lets you do really accurate single nucleotide variant calling. And so why do you need short reads if you can do everything with this? So that's what they're claiming. You can get the normal sort of variant calling, but also get all the phasing and the structural variation detection that you can get from the long read. So that also increases their data yield to 50 gigabases per smart cell, which is still much less than Illumina, but much more than it was before. So there's a future upgrade that they're talking about that they announced. I know they announced at the ASHG meeting. I don't know if I think there might have been a press release before that, but they're going to, basically using the same chemistry, they're going to have an 8M smart cell. So that's 8 million zero-mode waveguides, which will be, in theory, 8 times as much data. So that will sort of start enabling you to do a full 30x genome on their machine. Whether or not, we have to see that data, but that's sort of something that we're kind of looking towards and seeing if that's something we might want to invest in, is that upgrade. But yeah, so bottom line, we still have PacBio, yay. It's going to be kind of down for a few months, boo. But by early 2019, we will have it up and running. And then we hopefully will be able to offer these sort of new chemistry improvements and maybe open it up to more members of the community who might be interested in PacBio. Oh, yeah, and then Illumina is buying them, so that's interesting. They announced last week that Illumina is purchasing them for 190 billion or some ridiculous amount of money, right? It seems like no, that's too much money. 1.9, okay, whatever, a lot of money. I'm bad at money. So a lot of money. So that'll be interesting because they've been a competitor with Illumina up until now, but Illumina really doesn't have a solution for long reads. So we're hoping that you're not buying them to like sunset them, that they're buying them to sort of incorporate them and improve them and stuff like that. So we'll see how that goes. So yeah. So the other sort of long-read sequencing technology that's out there that's very popular with folks is Oxford Nanopore. So this is nanopore sequencing, and how this works is they've got their flow cells, or I think they're called flow cells. I'll show you in the next slide. Basically, their surface is covered with nanopore sort of protein assemblies here, and your DNA gets sort of dragged into the nanopore. And as it's sort of being pulled through the nanopore, the conformation change that happens for each base is recorded, and the sort of shape of the bases actually give a slightly different signal. So each base gives a slightly different signal, and so it can kind of tell which base is which. It sequences a little strangely. It doesn't sequence every single base. It kind of sequences sort of fits and starts of like every few bases, but if you sequence enough data, you can, in theory, piece everything back together. So their max read length is also 100,000 base pair. They have no amplification required, and they can do, so it's basically they can use native DNA or RNA. So they're basically taking the raw RNA and sequencing that, which most technologies can't do. Most technologies, you have to convert it to DNA somehow and then sequence it, PacBio and Illumina included. So that's kind of an interesting feature. And because it's quote-unquote native DNA, it can detect modified bases as well. So methylation, things like that. So that's sort of its kind of claim to fame. And they have a few different flavors as well. So the kind of original Oxford was this MinION, which is this little tiny self-contained flow cell here that, their whole thing is you just hook it up to your laptop and sequence. Anyone can sequence anywhere, anytime, sequencing for the people. So that was their thing. They've since kind of realized that people want to sequence maybe more than just a small tens of gigabases per run. So they've kind of beefed up their offerings. Now they have something called a GridION, which is if you run, I think it's five flow cells? Yeah, 5 flow cells, and if you run them all at the same time, you get 150 gigs. The PromethION is their big guy. That's got 48 flow cells, and if you run them all, you get thousands of gigabases, apparently, up to thousands of gigabases. But they're all kind of just variations on this. The guts of this just basically like tandem, daisy-chained all up. They do have on their website the smidgen, I don't know how you say this, but it hooks up to your phone. Like this is their whole thing, is that you can do sequencing anywhere, anytime. So this is in development, obviously. They say this on their website, but in theory, you just plug your thing right into your iPhone and you can sequence, which is kind of neat. So at the Broad, multiple groups have tried the MinIONs at the Broad. The microbial omics group actually has purchased one of these GridIONs. They want to use it for, obviously, their microbial sequencing. And the GP is actually is still sort of deciding our strategy. So by the end of the year, we'll sort of figure out if we're going to invest in this technology or just kind of stick with these sort of like one-off MinION systems. So. But, sort of just want to point out that for us at least, analysis is the main challenge for long-read technology. So, particularly in the data sciences platform at the Broad, folks, rightfully so, spend, 95% of their time thinking about Illumina data, right? So short read data, how to analyze that, call that, do all those things. they're planning on building out their long read technologies, but we're also looking for partners, researchers who have driver projects that they really want long reads for and that have bandwidth or expertise to help look at that data. Because I can plug in a MinION and run something on it and be like, yay, I got a fast queue. But like, what do I do with that? I don't know. All the analysis methods that we know of are very tuned towards Illumina sequencing. So I think to really sort of get the community going with long reads, we really need folks who are interested in kind of helping us drive that and using the data for real. I could sequence the genome in a bottle trio all day, but what is that really getting us? We want to sequence real projects and help people sort of look at the data and use the data. And so that's something like we're sort of actively kind of talking to folks about are long reads something that the Bro community as a whole is really into and really wants, and then how can we work together to get those methods? So our sort of genomics platform portfolio leads, I don't know, there's someone who Tara Bowers is sort of in charge of the germline kind of community. So she's been talking to a lot of PIs, and then Carrie Zabolskis is in charge of the sort of cancer portfolio. So we'll kind of have them be talking to folks over the next few months, trying to figure out how the best way to start working with these technologies are in the GP. So yeah, I finished up early. Sorry, I talk fast. But do you guys have questions or anything where we can, while we're set? Oh yes, Sinead.
Just to go back to something you mentioned at the beginning about the Illumina switch from PySeq to NovaSeq.
Yes.
You mentioned that ideally people should avoid mixing data types in joint calling sets, and inevitably we're going to be doing that a lot.
Right.
I think joint calling actually is probably not
Okay, maybe
as much of a concern as individual small cohort where you've started on X. I think the overall big joint calling that we'll do with hundreds of thousands of genomes will smooth out any technical artifacts. I think that we shouldn't be concerned about that.
Right. I think like Nomad and things like that, you just have to, like, as long as we're aware of what technology everything is, and we've had that with XACT2 with the exomes. Just, like, you can joint call them all together. You just have to sort of note what everything was done on. So yeah, I was more thinking like if you had 100 tumor samples that you wanted to look at, you ran them 50-50. That might be too small. Like,
yeah, honestly, I think the biggest impact would be if you were doing some more somatic kind of analyses. Like we've seen, because somatic analyses rely a lot more on modeling noise in the system, using panels of normal data to filter out that noise at low allele fraction variance. And we've seen in NovaSeq, just because of the two-color chemistry, that there is just different noise models. They can be filtered out, but you'll need different filters. And so mixing anything where you're trying to do very sensitive low allele fraction type things would be more concerning than standard SNP indel germline activities. If you're doing mitochondrial heteroplasmy, again, just maybe stick on one or the other. But
that was a very useful overview. There's a lot of people who are sort of have rare disease models. That's predominantly what the medical population has, and they're at varying stages. I say as director of clinical research at the MGH for 16 years, I watched this. There's a waves of people getting ready to start, and there's a lot more coming than you have seen. And so they're sitting here thinking the same way I am. All right, if I'm going to start now, what would you place your bet on if you were going to go forward with a cohort of people over time, over 1 or 2 years? When would you make a transfer to this? Would you wait till 2019? It's only a couple of months away to do some of the pack bios. Would you go with what you have now? So that's question number one. And question number two is this perennial question of data analysis. Between you guys and us guys, the ones that you're looking for help in the long reads, they're a terribly small minority of the population. And most of us need help with the analysis, and there is no easy, simple way to do this. So there needs to be a service that is a bridging service if you're going to empower groups who would have the populations and have the cohorts and have the phenotypes and have the DNA, in fact, to be able to use this technology. So it's everybody comes away with this thinking, gee, I wish I could do it, but I don't have the bridging technology. So what are your answers to both of those difficult questions? I understand.
So for the first part, I would suggest just based on even conversations that we have with both these companies, that the cost to say try to do all of how we traditionally have been doing projects in terms of like I want a genome or I want a deep coverage exome or something like that, the costs on these still aren't competitive yet. So it's more of sort of right now maybe an additive technology. So you start with your short reads, get your meat of your data, and then if you still don't figure out what you're trying to figure out, then we can add on these long reads to sort of help maybe figure that out. So that's kind of how we're initially kind of thinking of it. I think it's more of like an add-on. Eventually, if they make the technologies cheap enough, it might be that I just do my whole genome on here and see what it looks like. But that probably won't be for a while. So
Yeah.
So they're
They may never get
yeah.
there. Now that Illumina
they
bought
can-
them, I think
right.
it'll be. I don't think Illumina will sunset it. I think really Illumina is seeing, the value of a mixed long and short read data type, really to capture the entire catalog of variation in a human, right? I think that's the place where SNPs and indels are pretty easy at this point. Structure variation is still hard, and so really to capture that entire breadth, I think we'll end up seeing this hybrid model. And there is, and we have a couple of projects planned and pilots planned to look at sort of dosage of long and short reads to say, is 20x or 30x short reads and 5x of long reads the right recipe to get you, what you need. And then it does require the methods development. The Broad SV team is certainly actively engaged in this to do the second part, which is not just give people long reads and say good luck. It's really about certainly what we're interested in doing. I think I'm not sure if it's this group we've talked to in the past about sort of GPs generally moving towards trying to add more useful primary and secondary analysis to the data we deliver, not if people want it, not just raw data. And we have this translational analysis group who can do variant calling for folks now. And so there is a mechanism for, as DSP creates new methods, we'll start to incorporate them into more of the data products that are coming out. We started that with clinical genome, where SNPs and indels will be sort of part of that. Broad SV is creating a method for SV calling from short read technologies that will be part of the genome. Sarah Calvo and DSP methods worked on good methods for mitochondrial genome variant calling at varying levels of heteroplasmy. That will become part of it. We've got methods for repeat expansion calling. So these are we're now starting to add on methods to the data that we're producing and working with groups to maybe to do exactly that second part, to try to help with getting people more of the data ready for the biological insights and less having to worry about the bioinformatics crunching of the data.
There's a huge number of people trying to convert from whole exome sequencing to whole genome right now. There's a big wave there now. I mean, and those are all terrific methods, and I appreciate the way you've been incorporating them. What do they do now?
So I think as Maura said, I think the long reads are, I would put that as another add-on to say, but DNA short reads, I'd say. You're right, all the studies are sort of trying to figure out exome versus genome. And we should point out, we didn't talk about it here, exomes are not going away. And in fact, we'll probably come back again, maybe in the new year. We are switching our exome within GP. We've started talking to people about this, like just a different content, different vendor exome that is actually a more even exome. So it will come to your original question of cheaper, better quality exome. And actually the exome price will go down significantly. And so there are very large projects that will still do many, many, many exomes. And so that's certainly will still be an option. I think it always just comes back to the intended use. What do you want to do? Do you want to maximize for sensitivity for rare disease diagnostics, in which case you want to throw the kitchen sink at it because you've already done the standard of care gene tests, and you want to go and look, what could I get in the rest of the genome? And that's certainly what Sake's group at CGM is doing with a clinical utility study of the clinical genome, randomized trial of folks at different clinics in Mass General who are doing standard of care or standard of care plus a clinical genome, and understanding, would you have found everything that you would have got on the standard of care? Would you have found anything else? And so that's, those types of, we're in that phase now of utility studies, really depending on what you want to do. However, if you were doing the common disease, large scale, I'm looking for the noise and I need to do tens of thousands of samples, exome will still be competitive for a long time because numbers are the key, right?
As you look at the Mendelian community and rare diseases, there's a rapid consensus emerging that you get about 30 to 50% of your cases described by whole exome. But that means 60 to 70% of them, and that's the ones that are all moving to whole genome.
Yep.
Ideally, I mean, they have no idea what it's methylation or these other things, but the next logical step is there. And there you run into this huge analytic problem, just size of data, and the cloud helps study that.
Yep.
But the analysis is overwhelming to
Yep.
those people. And
Absolutely.
that's a huge, huge problem that you're creating, in essence, by being as good as you are. But in the wake of these, every time somebody comes here to ask them, that you're too busy to do this. And I understand that, it's the right answer, you know, but there is a shortfall there. There's a moat around the road, and there's few drawbridges to get in and out. And I think it's something that's worthy of attention. There's real opportunities for people to do consulting, for example, a group by group. I don't want to sheep steal and, you know, basically dilute the efforts for what you're doing, because it's terrific. But there may be opportunities for junior people, there may be a way to organize it, there may be a way to find new opportunities there that I find the road not necessarily considering because of the daunting face that you are facing forward in the future. But take a look in the rearview mirror, because there's a whole lot of disease alleviation of pain and suffering that can be done by just providing interpretation for whole genome sequencing for people that have assembled these cohorts and have done whole exomic sequencing. It's a real unmet need.
Yeah, absolutely. I actually think a lot about that. And I think, you know, again, I was at CGM yesterday where they were talking about this clinical utility study, and it did bring up the point of, like, if you wanted to do whole genome sequencing, even as a standard of care at some point, outside of an academic medical center or somewhere like this where you have all of those resources, it's almost impossible for somebody to stand up the resources to do it. We are thinking pretty actively, and we have a lot of sort of discussions about how, you know, right now we use the cloud for lots of things, but for most small projects, like you said, it's mainly a data delivery mechanism. But I think we want more and more to, like I said, bring in those methods and tools from the community and make them available in that cloud. So one, people don't have to download data, find storage for it, move it around, which is all expensive and bulky. They can do it there. And also, we have a lot of discussions with kind of the FireCloud team to build more interoperability between where we put our data and other systems outside the board that people already may have signed up for. So we go talk to different hospitals, and they're like, well, we've already kind of set up our clinical workflow on Bgi Nextcode. And, you know, they've done a lot of work on that, so wouldn't it be great if in the future they did a genome sequencing project here, they can find their FireCloud workspace and press a button and say, launch it in my other cloud-based analysis platform. There's a lot of technical work to be done there, but I do think that's the right way to move. So
that is in fact what we've done. We've gone to the Dana-Farber bioinformatics board that does fee-for-service. John Quackenbush put it in, and we download the data. It was a little hard to get the interfaces going with the core, but it's there. It's up in the cloud and everything. So, I mean, that's our one-up solution for our cohort, 1,600, very rare, world's largest cohort disease. But we're prototypical of a lot of things. The capacity exceeds the bandwidth.
Yep. So I think this is all great feedback, and it kind of validates what we've been thinking. I think anybody who has projects where, look, you guys are giving me data, but I need help on the informatics and it's not clear where to go, please do email us, genomics@Broad, or tag at Broad, or just email me. And we are all the time sort of building these analytical services that we can actually do in that sort of service-based model. And we really want to hear about what people want. We've had a lot more uptake on the cancer side, honestly, recently, from Dana-Farber and other groups, but I think we'd love to understand what people really want on the germline side as well. Any other wish lists of technologies, of data types, beyond it being cheaper and everything else, you know? What are the wish lists for the community? How important is RNA? Like, you know, we talk about the technologies, should do long reads, is it really important to do splice isoforms, or, just better RNA in general would be good? Folks have sort of thoughts? We always think RNA is very important to us, and clearly, obviously, with things like GTEx, there are some pilots on going into some GTEx samples and doing long reads to kind of characterize some of these things, but it's hard for us to understand where to put our efforts sometimes. Should we be looking at different types of RNA kits just to make RNA better or cheaper? is longer better? You know, so any thoughts, feedbacks, again, please sort of contact us, let us know.
Just to answer your question, I work as an analyst in the rare disease space, and there are often, since we take in samples that have been pre-screened, so we're typically looking for, answers that previously not found, and I often come across like cryptic splice sites or synonymous, we don't know whether it's affecting splicing at all. So if there's a better, more efficient way for us, if we could run RNA-seq and see if there's like some sort of effect versus just guessing and saying, current splice predictors aren't predicting anything, so we move on. But if there's a fraction of it there that we can elucidate by running some of these RNA-seq experiments, I think that would be really great.
Okay, good.
Brain data.
Right. Good.
Could you speak up when you hear that?
For brain data,
Yeah.
it's all about splicing.
All about what?
A lot of it is about splicing.
Yeah.
Splicing. That's hard.
We're tracking the GTEx data, tissue-specific GTEX data, and QLs is the way to, one way to get at that, because there's a lot of variability cell by cell, but that's kind of the normative data. We don't know if we're looking at the canonical sequence, is the right one, etc. Splicing is a really big deal in the brain.
Right.
And that's all mainly sort of shorter insert data as well. So you might not see necessarily if things are only a couple hundred base pairs long. So that sort of argues longer may be better for RNA. Yeah.
Yep. One final closing thought. I just Maura mentioned that we were looking at people are looking at alternate service providers for genomes and things outside that appear to be much cheaper. I think most folks will know. I mean, the GP, the Broad, has been awarded a sequencing center, or we are a sequencing center for the All of Us project. NIH, in their wisdom, like to distribute things. So there's actually 3 sequencing centers: us, Baylor, and UWash. Each of the groups will do a third of the genotyping arrays, which every All of Us participant will get first. So there's a custom array for the All of Us project, which is an Infinium array, 1.8 million sites, which takes all of their sort of previous GSA array, their multi-ethnic global array, and just bolstered all of the pharmacogenomics and ACMG content. That array, we have it in our hands. We'll be running a validation of it now. It will be available outside of the All of Us project. So if anybody is interested, that will be a relatively cost-efficient array that will be available for anybody. So each of the genome centers is doing roughly a third of the arrays. That's 150,000 total next year, so 50,000 at Broad. And then, at least in the next year, Broad will be doing 60% of the whole genomes. So ultimately, over the next 5 to 6 years, this will be 1 million people characterized. Everyone will get an array, and hopefully everyone will have a genome. And regardless of where the data is generated, Broad is also the data research platform for All of Us, so that all the data from all the centers will come here. It's us, Verily, and Vanderbilt is the DRC, the Data Research Center. So as you think about applying to access the data in the future, most likely we'll take in all the genomes from everybody and do recalling, a joint call set on everybody. So this will become a fantastic resource for research. If you send your genomes out and get it done on a MegaSeq or a Bgi Seq, the data is going to look different. And so you won't necessarily be able to analyze it in the context of this 1 million person cohort. So just think about 5 years from now, if you want to do a population study and look at frequencies and everything else and analyze your data in the context of a 1 million person cohort, that data type will be really hard to work with. So just putting it out there again is kind of a think for the future. All right, I think we're probably good. Give people a moment to catch breath before the next session. But again, please, if you haven't had your groups come to 320, we do tours every single day, every week. Please bring your students over, come over yourself, just send an email. We love to host tours. Yeah. Thank you, everyone.